a difficult major. Admission to science majors is very important to the future of the country and the world. So it's very good what you're doing. There are a lot of problems that are going to need to be solved if we want to continue as a human race, if we want the planet to continue as a livable planet. So there's so much to be done. How many of you want to be researchers? A lot of you. Clinicians? Teachers? Industry? Entrepreneurs? Yeah, very important, very important. Yeah. So we need to find new ways to prevent and treat disease. Renewable energy. How are we going to keep power going in the future without consuming food resources? Healthy planet. Green and sustainable industries. Sustainable agriculture. Economic growth. The U.S. economy is not going to grow on mature industries because other countries can make things cheaper than we can for mature industries. We need to build the economy on innovative technologies, and that depends on you in the future. Invent new things to take to market. If it's new, if it's innovative, there's less competition, and that's where the future of the U.S. economy is, in my opinion. So good for you. Start off, before I start talking about me, I wanted to talk a little bit about you. Okay, so here's how this talk is organized. A little bit about me, about structural genomics. There are over 14,000 protein structures of unknown function or uncertain function in the protein data bank that were discovered as part of protein structure initiatives in the last 18 years. I'll talk about protein electrostatics and our electrostatics-based methods for finding functional sites in proteins and how we use that to predict function. So that's what I'll talk about today. Okay. So, Seikon, Mary Jo Ondrechen. That's how I looked when I was in college. I'm a member of the Ganawagi Mohawk band. I'm a half-blood. So I just said, hello, my name is Mary Jo in Mohawk. So that's how I looked when I was where you are now. This is me at 2 months with my mom and dad. My mom did something very important for me. She told me that I could do whatever I wanted if I worked. If I tried, if I worked, I could do whatever I wanted. So even though I got bad messages from other people, like, you know, as a woman, you're never going to make it in science. As an Indian, you're never going to make it. You're stupid. You know, you're not good enough to major in science. I got those messages. But my mom told me over and over and over again that if I wanted something, if I wanted to do something, I could do it if I worked. So that was a very, very important message. You know, that you, in the future, you may have kids. If you tell them over and over and over again, you can do whatever you want if you work, if you try. You just keep telling them, they'll believe you. Okay? So that was some very important thing that my mom did. Okay, so these are my grandparents, Stephen Oroniateka and Bianca Scavanetsi on the Ganawagi Mohawk Reservation. They were trilingual. They spoke Mohawk, English, and French. I never knew my grandfather because, like a lot of Iroquois men, he was an iron worker, and when my mom was a small child, he was killed in a high-level fall. So I never knew this grandfather. My grandmother was a historian. She wrote articles about the history of the local region. But in those days, women were not allowed to publish. So a man would take her writings, put his name on it, and publish it. So her work was published under someone else's name. So now just to be able to publish articles in my own name is an improvement over the way things were. Okay. Okay, so how did I get here? The teachings of my elders first. I went to public schools. I got a high school diploma. The public school I went to was the kind of school where you don't learn very much, so I wasn't particularly well prepared for college. I got a scholarship to Reed College at Portland, Oregon. It's a small liberal arts college. It's co-educational, but liberal arts like Wellesley. And I majored in chemistry, and then I got into the PhD program at Northwestern in Illinois. I was a NATO postdoctoral fellow in Israel. And then I came back to the US in 1980 to join the faculty here in Massachusetts at Northeastern University in Boston. So I've had the opportunity to work at other places. I've had visiting appointments, sabbatical appointments in Aarhus, Denmark, University of California, San Diego, for Andice and Scripps La Jolla, and then later at Caltech. So I've had the opportunity to work at other places. That's one of the great things about being a professor, because you get sabbaticals and then you can go to other places and learn new things. So it's a very, very nice life. Okay, so a little bit about genomics. Big promise, big challenges. So the human genome was sequenced. Well, you guys were born into a genomics world. Okay, so the human genome was sequenced right around the turn of the millennium, and hundreds of other species were sequenced later. And it was said that, okay, if we can sequence the human genome, we'll know everything there is to know about disease and human health, and we'll figure everything out. Well, you know, that was a little ambitious. You know, the genome was sequenced, and then there was this big pile of information, and it was not so easy to interpret it. So, yeah, late 1990s, 2000, human genome sequenced. Thousands of other species have now been sequenced. All kinds of bacteria, plants, animals, you know, cats, dogs, rats, kangaroos. All kinds of species have had their genome sequenced. The structural genomics effort. Okay, so it says, oh well, we know the sequences of all these genes. So if we could get the structures of the proteins they code for, then we'll know everything there is to know. Okay. So the U.S. spent about $1 billion on the structural genomic, on the protein structure initiative. There are now over 14,000 protein structures in the protein data bank, and nearly all of these are unknown or uncertain biochemical function. So just because we know the shape of the protein doesn't mean that we know what it does. Okay. So the next frontier is functional genomics, trying to figure out the function of these protein structures. Okay, so some common approaches to biochemical function prediction are sequence comparison. You could do a blast and compare the sequence of your protein with the sequence of proteins of known function. And if they're similar, you can say, well, maybe they have the same function. Evolutionary history. You could construct a phylogenetic tree, match the function to those of neighboring proteins in the tree, or you could compare the 3D structures. So these are informatics-based approaches. And you know, around the turn of the millennium, bioinformatics was an emerging field. And just about everybody in it was either a biologist or a computer scientist. And that's fine. And I said, well, gee, you know, they really ought to have some chemistry in it. So that's when I stepped in. I saw an opportunity, I stepped in. We ought to have some chemistry. Let's add some physics and chemistry and see if we can learn a little more. Okay. Okay, so the original planning end of the structural genomics effort was that, okay, if we know the 3D structure, we'll know what these proteins do. Okay. It's much more difficult than anticipated, and therefore there's a high rate of functional misannotations. There are databases where over half the proteins in the database are expected to be misannotated. They have the wrong function label because the functions were assigned by the closest sequence comparison or the closest structure comparison, and that doesn't always work. Okay, so we need a new approach to function prediction to identify targets, you know, targets for antibiotics, targets for human diseases, and to add biological knowledge to the structural genomics data. Okay, so that's where chemistry comes in. Okay, yeah, just because we know the structure, of course, we don't know the function. This is the Rossmann fold. You guys have all taken biochemistry, is that right? Everybody's taken bio? No. Okay, oh, well, there are lots of possible ways that proteins could fold into three-dimensional structures. Globular proteins tend to have a pretty fixed structure. They're dynamic, but they have a sort of same overall, the protein stays in a particular fold. So this is the Rossmann fold. And this is the tin barrel. It's got 8 beta sheets surrounded by 8 alpha helices. I think that's a really cool structure. And Each one of these folds represents over 50 different biochemical functions. So just because we know the structure does not mean we know the function. Okay, so our approach is to use computed chemical properties of individual amino acids in the protein structure, combined with evolutionary and geometric data, and identify the amino acids that are important for biochemical function, and then seek a local structure match, not a global structure match, but a local structure match at the part of the protein structure that does the biochemistry. So that's the way that we predict function. Okay, so first we find the local site where function occurs. And we have developed methodology to do that. If you give me the structure of any protein, if it's an enzyme, I'll tell you where the active site is. And if it's a small molecule binder, I'll tell you where that small molecule binds. Okay? All based on computed electrostatics and chemical data. All I need is the structure of that one protein. I don't need to compare with other structures. Okay, so now, do you guys know this, right? That 7 out of the 20 natural amino acids can have an electrical charge, right? Histidine sometimes can be positive. Lysine and arginine are usually positive. They can also be neutral. The aspartate and glutamate are organic acids, and so they could give up their proton and make the carboxylate anion. Cysteine sometimes can be negatively charged because it has a thiol group. It can give up a proton and make the thiolate. And tyrosine is a phenol. It can sometimes give up a proton and make the phenolate. So these could sometimes be negatively charged. Aspartate and glutamate are very often negatively charged. These guys could be positive. So 7 out of the 20 natural amino acids can have an electrical charge going from neutral to charged. Oops. Yeah. Okay, so where is the reactive part of a protein structure? We use computed electrostatic properties, calculate the electrical potential of this. We represent the protein as an array of charges. We solve the Poisson-Boltzmann equations to get the electrical potential function. So you know, physics 101, if I have plus charge here and a minus charge here, and I know the distance between, I can sit down and calculate the potential function. All right, if I have 4 plus charges in a perfect square, and I know the distance between them, by the symmetry of the system, I can sit down and calculate the electrical potential function. But if I have plus and minus charges all over the place, then I can't solve for the potential function exactly, but I can solve in pretty good approximation numerically on my computer. So that's what we do. We solve the Poisson-Boltzmann equations approximately, obtain the electrical potential function, and use that potential function to compute chemical properties. Okay, so we invented a method. We worked with Dagmar Ringe at Brandeis, just up the street, and we made a method called theoretical microscopic anomalous titration curve shapes, mathematics. What we do is we calculate the electrical potential of the protein, and then we calculate the theoretical titration curve of all these 7 amino acids that are able to exchange protons. And so 75% of all catalytic residues are among these 7, or the NC term, this can also be charged. Okay? And so here's what we do. Okay, we calculate the theoretical titration curve. So if I have a large ensemble of protein molecules, I can have, I calculate the average charge on a given amino acid. So this is the average charge on a given amino acid in a large ensemble of protein molecules as a function of pH. So this is kind of like the titrations that you did in Gen Chem, except it's turned 90 degrees on its side, right? Okay, so this is the midpoint of the titration curve where the net charge is 0.5, okay? And then the equivalence occurs down here when everybody is converted. So like, you remember, like if you have they give you an unknown acetic acid and you have NaOH in your burette and you know the NaOH concentration, you start adding and adding and adding, and you put phenolphthalein in and it's clear. And then You put one drop and you get that endpoint that's just faint pink. And then if you overshoot and get one more drop, it turns, you know, this very, very bright pink. So that's the equivalence, and that's down in here. Okay, all right. And these are all done on computer. To actually titrate individual amino acids in a protein is a very expensive, labor-intensive process. The only way to do it is on the computer. Okay. So these are selected lysines, and you know, lysines have that primary amine group. For those who haven't had biochemistry yet, lysines have a primary amine, and so they can be positive or neutral. Beta-lactamase is an enzyme that bacteria use to foil antibiotics like penicillin. Okay, they will destroy the antibiotic. Yeah, bacteria are really, really smart at figuring out how to foil antibiotics. And antibiotics-resistant infections are getting more and more common, especially in hospitals. Okay, so if we take a look at these curves, you notice that these two with the dark symbols have the sigmoidal curve of Henderson and Hasselbalch, right? Whereas lysine 73 and 234, the white symbols, they have elongated titration curves where you have partial proteination over a very wide pH range, and those are the two that are in the active site. Okay. Same over here. These are selected glutamates from arginine kinase. And so you don't have to know any biochemistry to say, aha, this one right here, GLUT225 is different, so this is the one that's in the active site. It's got this elongated titration curve. Right? Okay, and we want that, right? Because by definition of a catalyst, the catalyst has to regenerate itself for the next turnover cycle. The catalyst has no net change in one cycle, right? So if it donates a proton, it's got to take a proton back. So it's got to be able to go both ways. So we need a wide buffer range, a wide pH range over which both protonation states, charged and uncharged, are populated. Protonated, deprotonated are populated, because if we get out in the asymptotes, the reaction doesn't go. Okay, so we want to have both protonation states existing over a wide pH range, because proteins are dynamic. They sort of vibrate a little bit. They have ions drifting in and out. One small change, if you have a sharp titration curve like these guys, one small change could shift you into the asymptote. And you don't want that. Okay, so in order to be active, you know, Mother Nature makes all these coupled protonation states so that the active residues are able to be protonated, deprotonated both over a wide pH range. Okay. So this is just a polyprotic acid effect. You know, in organic, you learn your diacids, you know, oxalic. My memory's a little rusty. Okay, how does it go? It goes, oxalic is with 2 carbons, right? 3 carbons is what? the diacetyl, you know, oxalic, malonic. It's a polyprotic acid effect. You have coupled protonation events, coupled proton transfer reactions. Okay. So the active amino acids are coupled to neighboring amino acids to give these anomalous titration curves. So we tested the method on a database of curated enzymes with a woman named Gail Bartlett in England wrote her PhD thesis combing the literature for experimental evidence that particular amino acids are involved in the catalytic reaction. And then she compiled that into the catalytic site atlas. And so that was the database that we used to benchmark the method. That was her PhD thesis, just to make this database. Okay, so then we wanted to extract more information from these computed electrostatic properties, so we invented a new machine learning method called pool. Now, you guys know what machine learning is, right? That's where you have a bunch of labeled examples, and then the machine learns to sort the examples, and then you give it an unknown case and it sorts for you. Like I think a lot of the spam filters in email try to work this way, right? You tell the filter, okay, this is good, this is spam, this is good, this is spam. And then it sees the characteristics of your good messages and your bad messages, and then it takes that information and it learns, so if an email comes in, it can figure out with some degree of accuracy, some are better than others, whether it should be classified as good mail or spam. Okay, so that's machine learning, where the computer learns. You give it examples, it learns from those examples to classify, and then it classifies based on the set of learned examples. Okay, so pool is a multidimensional isotonic regression with a monotonicity constraint. So it's a regression, but it has the constraint that the outcome has to be a monotonic function of the input. Okay, so our metrics from those titration curves, the bigger those metrics are, the bigger the deviation from Henderson-Hasselbalch sigmoidal behavior, the bigger the deviation, the more likely that amino acid is involved in catalysis. Like you say, the higher the conservation score, the more likely it is to be involved in catalysis. The more it's in a concave part of the surface as opposed to a convex part of the surface, the more likely it is involved in catalysis. So you could invent features where the outcome is a monotonic function of the feature. So we took our metrics from those titration curves, and we also added environmental features that describe the electrostatic surroundings of all amino acids. So now we can predict all 20 amino acid types, not just those 7 ionizable ones. And we get very good performance in predicting the amino acids involved in catalysis. Okay, so that's how pool works. Okay, so we have two methods then, thematics and pool, that predict, if you give us the structure of any protein, we'll predict where the catalysis happens, where small molecules combined in that structure. Okay, so here is a picture of methyl glyoxal synthase, and the reds are the predicted amino acids, and this is exactly where the catalysis happens. These are the annotated important amino acids. So it's not the whole structure, it's just this one part of the structure where the actual biochemistry happens. So that's the kind of thing. We predict sets of amino acids that form typically a cluster in space, and that's where the biochemistry happens. Okay, so we know we can predict functionally important amino acids. Does that mean we can take all those structural genomics proteins and predict their function? So that's a jump to go from predicting the functional sites to predicting the function. Okay, so our approach is to take proteins of known function and predict the functionally important residues, and then say, okay, if I have a set of proteins of common function, and they catalyze the exact same biochemical reaction, what do their local catalytic sites have in common? So we predict the important amino acids, and then we say, of all the proteins with this particular function, which of these predicted residues are common to most of them? And then that gives us a local motif that is characteristic of that function. And then we define then functional subclasses of proteins with these local signatures, these local motifs. And then for proteins of unknown function, we do the same prediction of the important amino acids, and we compare them to these local motifs and see whether we get a good local structure match. So we're not doing a full structure match, we're just doing a match at that local site of activity. Okay, so this is the ribulose phosphate binding barrel superfamily. It's got the Tim barrel fold, and its enzymes do all kinds of important metabolic activities like amino acid synthesis and things like that. Okay, and these are the known subclasses, the known functional subclasses. Like tryptophan synthase makes tryptophan. Some of these are decarboxylases, epimerases, ligases, all different kinds of activity in this set of proteins of common structure. So they all have the same fold, they have the same structure, but there are these branches that do different biochemistry. So we want to be able to identify which ones, how could we identify which local sites are characteristic of what activity? Okay, so what we did was, this was Jocelyn Lee's PhD dissertation. She took the proteins of known function in this superfamily, and she calculated the important amino acids, and then she did a structural alignment and said, okay, well, look, each of these functional families has a unique set of important residues in particular spatial positions. So here, in this table, each row is a protein structure. And the ones of common function are grouped together. And each column is a spatially aligned position. So we superimpose the structures on top of each other, and then we paint with a paintbrush the amino acids that we predict to be important. And then we say, ones of common function, what do they have in common? These painted amino acids, all right? And there's a unique signature. The blue highlights are the signature residues for each functional type, and they tell us then what does a tryptophan synthase look like. So what does a ribulose phosphate epimerase look like? Well, the local site has these features. And then we have a scoring function to score the unknowns against these knowns. Okay, so the chemical signatures are unique for each functional subgroup, characterized by residue types that we predict in particular spatial positions. And so we can make a little vector like this. So a phosphoribosyl anthranilate isomerase has a cysteine in position 1, a glycine in 3, arginine in 8, histidine in 14, like this, okay? And this is unique, all are unique to this particular family except for that H14. Okay, so we have a unique local signature for each functional type. Okay, and so this is a picture of the alignment. This is a picture of the alignment of the known RPEs and the structural genomics proteins that could be RPEs. And we see we get a pretty good alignment. So that's how it works. We score them. Okay, here's another example. The 6-hairpin glycosidase superfamily is an all-alpha structure. It has 4 known functional subgroups. Ramya Parasuram, this is part of her PhD thesis. This is what she predicts for the functional residues for the glucoamylase family. This is how they look for the knowns. Okay. This was a putative glucoamylase from structural genomics. It belongs to a species called Bacteroides fragilis. That's a bacterium that we have a symbiotic relationship with. Okay. So this was a protein of unknown function from structural genomics, and said, this is a glucoamylase. Well, probably not, because the reds are the known glucoamylases, the blues are what we predict for the structural genomics protein, and we could not find any way to fold the structural genomics protein to match these guys. We say it doesn't have the signature of a glucoamylase. It cannot be a glucoamylase. And this is one of the thousands, about tens of thousands of misannotated proteins in the database. We can predict substrate specificity. This was a protein from Bacillus halidurans that was annotated as a fucosidase by the bioinformatics methods. But you see there's a mismatch here. Okay, the blue is the unknown, the red is a galactosidase, and the orange is the fucosidase. But there's a mismatch here and a mismatch here. So we said, gee, we get a much better match with the galactosidase. So we predict galactosidase activity because there's no room for the fucosidase to fit in that catalytic site. We get a much better match with the known galactosidases. Okay, these are the two putative phenylyl-CoA hybridases from structure genomics. In both pictures, the structure genomics protein of uncertain function is in magenta, and a representative known phenylyl-CoA hybridase is in green. Okay, this one is correctly annotated. This is one from Geobacillus caustophilus. We get a beautiful match with the signature. Okay, I just put one known up here because otherwise it gets too busy. But you see, we get a beautiful match of the predicted residues of the unknown with the known phenylyl-CoA hybridase. But look over here. This one is from Bacillus holodurans, also a putative phenylyl-CoA hybridase. The whole family has this CBK motif, but everything else is wrong. So we say, no, this is not an phenylyl-CoA hybridase. We think we know what it is, and we have experiments ongoing to test. So that's one that's incorrectly annotated. Okay, so we have some examples where we verified our predictions experimentally. Experiments take longer than our predictions. So we have a few examples where we verified with experiments. This is a protein from Bifidobacterium adolescentis, which is another bacterium that we have a symbiotic relationship with. And YP190028.1 comes from a domain of unknown function in the genome of Bifidobacterium adolescentis. The structure was solved by the Joint Center for Structural Genomics in California. The closest match by bioinformatics methods was DNA polymerase III. So the crystallographer that solved the structure, I was on sabbatical at Scripps at the time, and the crystallographer said, now this is a DNA polymerase III, I'm going to submit it to the PDB as a DNA polymerase III, but take a look at it. Because in the phylogenetic tree, it falls among a whole bunch of DNA polymerases. And the match was not bad with DNA polymerase III, but there's a mismatch here and a mismatch here. This looks like a metal binding motif here, but there's an H for C and a T for Y substitution here. So we said, gee, let's look a little further. So we looked at some more examples and we said, we get a beautiful match with histidinol phosphate phosphatase. We think this is really a phosphatase, a metal-dependent phosphoesterase. So the crystallographer believed us and submitted it to the protein data bank as a metal-dependent phosphoesterase, phosphatase. If you've had biochemistry in the lab, you probably did a phosphatase catalyzed reaction. It just takes the phosphate group off things. Okay. So subsequently, we made a test and showed in the lab that it does have phosphatase activity. My colleague Penny Boyning, who works on DNA polymerases, could not find any polymerase or DNA binding activity, so we were right. And the bioinformatics, in this case, was wrong, because chemical information can tell you more than just the informatics. Okay. Here's another example. Two putative ketosteroid isomerases. One from Mycobacterium tuberculosis, you know what that does, that makes people very sick. And there's another one from Mesorhizobium loti. Mesorhizobium loti is a bacterium that lives in the roots of certain plants, and it's involved with nitrogen fixation, so it's important for agriculture. Okay, so we compared these putative ketosteroid isomerases with two known ketosteroid isomerases that, and we we showed, yeah, that beautiful, beautiful structural match, perfect structural match. They both look, those two unknowns look just like the known key stored isomerase in the overall structure. But if you look at the local structure, the one from tuberculosis has a good match in the isomerase part of the active site. Not so great in the steroid recognition part, but it's clearly an isomerase. It's got all the isomerase amino acids. The nasal-rhizobium loti doesn't have the proper isomerase amino acids, although it does have a steroid binding pocket. So we said, we went out on a limb and we published. We said, no way nasal-rhizobium loti is the key to steroid isomerase, but the Mycobacteria tuberculosis one is an isomerase. Okay. Subsequently, experiments were done. Okay, and this was part of Srinivas Somaropu's thesis. By kinetics assay spectrophotometrically at 248 nanometers, we measured the conversion of 5-androstenedione 317 dione to 4-androstenedione 317 dione, and showed that the tuberculosis one is indeed an isomerase. The nasal-rhizobium loti has no detectable activity. You know about androstenedione dione, right? That's what the baseball players were using to hit all those home runs. Yeah. Okay. So we were right, that the tuberculosis one is indeed an isomerase, the other one is not. Okay. The crotonase superfamily has a lot of protein structures that have been solved as a part of the structural genomics project. There are 5 known subfamilies, and we predict that about 3/4 of them are misannotated. Okay, and the experimental data is not published yet, but we have correctly identified the isomerase activity of ones that we predict are in fact isomerases and not proteinases. Okay, so I'll wrap it up here. Okay, so our methods predict the active residues. The local site predictions are structurally aligned. Matching functions are detected using a scoring function at the predicted positions, only in the local part of the structure. We find the signature positions in the 3D structure and sort related structures into functional groups. And we annotate function of structural genomics proteins, identifying misannotations. So the message is that chemistry and physics have important roles to play in the interpretation of genomic information. Okay, I'll just mention our other projects. When we were benchmarking our method for predicting these local sites of activity, our methods for almost all enzymes predict amino acids that are not close to the site of reaction. They might be in the 2nd or 3rd layer behind the active residues. And so when we were benchmarking the method, we marked those as false positives, but we were wondering, are these false positives really false? Well, most of them are not false positives. Most of them really do something. So we have all kinds of experiments showing that if we mutate away these distant residues conservatively, we lose activity. They are important. They are contributing from a distance. And then we're using that knowledge for enzyme engineering. Enzyme engineering is a brand new field. No best practices are established yet. Typically, the computational predictions of enzymes that will catalyze a novel reaction, these are done, you might predict, make 100 sequences and test them, and maybe a handful of them will have teeny activity, 0.00001 per second per minute. Then you go in the lab, and if you spend a few years doing directed evolution, and after many rounds of directed evolution and a few years have gone by, you might have a decent enzyme. So that's not a very good way. That's trial and error, right? But directed evolution is really trial and error. So we want to build the right properties in early on. So the vision is to convert the chemical industry to enzyme catalyzed processes instead of thermal processes or conventional catalysis. Because You can save energy, you have fewer unwanted byproducts because enzymes are so specific. Problem is, most industrial chemical reactions don't have a natural enzyme known that will catalyze the reaction, so you have to invent a whole new enzyme. And that's a real challenge. So that's what we're trying to do. That's one of our other efforts. Okay, drug discovery. I have 6 medicinal chemistry colleagues, and we're their computational support. We help them design molecules. We have a grant from the ALS Association. Remember what the ice bucket challenge, when people were dumping those buckets of ice on their heads in the summer, not now. And did any of you do that? Anybody do that? You did? Oh, good for you. Yeah, because all the money that was raised, in the first year, it was $115 million. So the ALS Association issued this call for proposals, and we submitted one with this new idea, new approach to treat certain kinds of inherited ALS, and we got the money. So we're working on that now. We were working on a cardiac drug with one of our colleagues. We're working on anti-cancer drugs with another colleague. I mean, they do the hard job of making the molecules, and the biologists test them. We just do the computation to help them tweak the molecules to make them good binders to the target. Okay, so that's kind of a quick overview of what me and my group do. Okay, so here's some of the people that did the work. Now Dr. Ramya Parasaram, she did the 6 hairpin glycosidases, and she got a Best Poster Award at the Protein Society. This is Dr. Jocelyn Lee, who did the ribulose phosphate binding barrel superfamily, and she was an NSF-GRFP fellow. This is Caitlin Mills. She's doing all the experiments now on the proteinase superfamily, testing if our predictions are right. And in 2015 and 2017, she got Best Poster prizes from the Protein Society Symposium, and also is a former Foundation Fellow. This is Lydia Ruffner, who recently joined the group. She's working on the haloacid dehalogenase superfamily, testing our predictions with her biochemical assays. There are dehalogenases in that family which can be used for environmental remediations. So those are a lot of important molecules. These are some students who worked. This is Dr. Kobian, who worked on the proteinases. Mong Mary Touch wrote about the HAD superfamily, the haloacid dehalogenase superfamily in the master's thesis. Eve and Saffron were undergraduate researchers in the summer. Okay, so our work is funded by the National Science Foundation, the Pharma Foundation, the National Institute of Justice, MathWorks, and the ALS Association, and all those wonderful people who did the Ice Bucket Challenge. These are my current group members, former group members, collaborators, former students, so here's where you can find me. You can find me on Facebook and LinkedIn and Twitter, and the group, that's our group handle on Twitter. And again, thank you very much for your kind attention. I'm happy to answer questions.
Yeah. I just wanted to know what the main reasons were for why the bioinformatics approach for finding functional sites worked bad.
Oh, not finding functional sites. They compare the overall sequence or the overall structure. And the reason why it doesn't always work to it's called transferring. You get a similar sequence, you say, okay, it has the same function. You can have high sequence similarity and still have different function. And you can have very high structural similarity and still have different functions. So because there are similar structures with different functions, similar sequences with different functions, bioinformatics doesn't always give you enough information. And then once one protein gets misannotated in the database, then everything that's referenced to that also gets misannotated. So then there's a cascading error issue where one mistake can propagate into hundreds, thousands of mistakes. because a lot of the annotations are done by automated methods. So if something gets labeled with a certain function and it's wrong, then the automated methods are going to put that same label on a lot of other wrong things. Now, bioinformatics is very useful, but our argument is you've got to have some chemistry. Yeah?
Wait, if you could give advice to yourself as an undergrad, what would you say?
Oh boy. Well, I was very naive and very shy, and in those days, you know, a lot of the professors didn't take female students seriously. So I didn't find good mentors until graduate school. I happened to stumble into a fellow by the name of Mark Ratner. He's retiring now, but he's in the National Academy of Science. He's done very well. And he didn't mind having a woman in his group. He treated me the same. When his son was born, he passed out cigars and he gave me one, and he treated me the same, you know. So I mean, if I could give advice to myself, it was to seek out, find a decent mentor, because they're not all bad, you know. You've got to find a decent mentor. Here at Wellesley, it's probably easier because you've got women on the faculty, right? And you've got, and you got, I suppose there are men on the faculty, but they're more open-minded men, right? So you could, yeah, find a mentor, get advice from people. Don't be shy. Yeah.
So for all those local alignments, I could definitely see if they're a perfect match, then it's probably a very good predictor of them having similar function. But what if it wasn't? So there are some cases where they're imperfect matches.
Right.
So what if it was just a matter of like one of the residues and it's still say a residue that has the same, like if it was instead of arginine like a lysine, like they still have a positive charge is similar to decay. Like if it could still be a possibility even if it's an imperfect match and we still have similar function.
Good question. Okay, what we do is we have a scoring matrix. And so what we do is we score among the knowns. So we have a signature that comes from basically majority vote of the ones of common known function. And then we score the knowns against the signature. So if all the knowns are, you know, in the 0.90% range of this, you know, the scoring goes from negative to 1.0. 1.0 is a perfect match. Okay, so if the range of values of the scoring matrix element is 0.9 or better, then we expect that an unknown has to have 0.9 or better to be classified in that group. So we look at the range of the scoring results for the knowns. If the score, there are some families where there's more variability, and then if it goes like from 0.7 to 1.0, and we say, okay, if the unknown is in that range, 0.7 to 1.0. So to answer your question, we look at the range of scores within among the knowns and see if the unknown fits in that range. Other questions?
All right, thank you so much.
Oh, thank you, thank you.
You guys want to have more pizza? We're just happy to answer questions more personally.
Thank you.
Yeah, I'm happy to answer questions.